Welcome to Analytica Chemistry Vision, a platform dedicated to exploring the theoretical and experimental aspects of chemistry, blending classroom learning with laboratory research to enhance understanding and inspire innovation in chemical science. Let's explore our new experimental video on the determination of protein in food samples using the simple Gjeldahl method. You can also watch other videos on this channel covering fat, moisture, ash, iron, copper, and nickel determination in food samples. These experiments are highly used for UG or PG students, or research for all science discipline which can be performed limited resources or remote areas. First, here we see what is the principle behind the determination of protein from food samples. For protein estimation, when the amount of protein is percentage, then Gjeldahl method is used. This is a classical technique used to determine the nitrogen and protein percentage in organic samples. The sample is first digested with concentrated sulfuric acid in the presence of a catalyst, which converts organic nitrogen into ammonium sulfate. After digestion, the solution is made alkaline with sodium hydroxide, releasing ammonia gas. The liberated ammonia is distilled and absorbed in a known volume of standard acid solution. The amount of ammonia, and thus nitrogen, is determined by titration. The nitrogen content is then multiplied by a factor of 6.25 to calculate the protein percentage accurately. Here, we will see what is reaction mechanism for determination of protein from food sample. Organic nitrogen in the protein-containing samples is oxidatively by concentrated sulfuric acid, in presence of heat and catalysts such as potassium sulfate, and copper sulfate. Carbon and hydrogen are oxidized. Nitrogen is converted to ammonium ion, NH, which pairs with sulfate to give ammonium sulfate. Next, after digestion, the acidic mixture is diluted and made strongly alkaline by adding excess sodium hydroxide, NaOH. Ammonium is converted to ammonia gas, which is steam distilled and captured in a trapping solution that is boric acid. After, protein is estimated by multiplying percentage nitrogen by a conversion factor, commonly 6.25 for many food proteins, because average protein contains 16% nitrogen. For performing experiment, here we will see what are requirements. First, the reagents needed are copper sulfate, hydrochloric acid, boric acid, sulfuric acid, sodium hydroxide, potassium sulfate, methyl red, bromocazole green. Apparatus required for performing the experiments are Gjeldahl apparatus, heating mantle, flask, beaker, measuring cylinder, weighing balance, round bottom flask, burette. Here, we will see what our solution is prepared prior to start the experiment, which are given below. 5% copper sulfate, which is prepared by dissolving 0.5 gram in 10 ml double distilled water. Second one is 0.1 normal HCl solution for titration. Next is 40% borax solution, which is prepared by dissolving the gram of substance in 100 ml of distilled water. Then bromocazole green is prepared by dissolving the 0.016 gram in 100 ml ethanol, and methyled by dissolving the 0.032 gram in 100 ml ethanol. The indicator is prepared by mixing 1:1 of bromocazole green indicator and methyl red, used during the titration with HCl. As we know that food is good sources of protein. Proteins are vital for body growth, tissue repair, and enzyme and hormone production. Major vegetarian sources include milk, pulses, lentils, soybeans, nuts, seeds, fruits, and whole grains. First part of determination is collection of sample, and dried sample is ground into fine powder with the help of pestle and mortar or some other grinding machine. After, 1.0 gram amount of fine powder is weighed for measurement of protein in food sample using digital balance shown here. In the second part of procedure, 1 gram of fine powdered sample is transferred into the round bottom flask, followed by the addition 2 gram of potassium sulfate and 0.2 ml of 5% copper sulfate. The solution mixture is well mixed with shaking. And then 10 ml of sulfuric acid in the flask for oxidizing the food materials, and shake the solution mixture very carefully. And during the experiments, the lab security precaution should be considered. After the addition of sulfuric acid, the glass connector is joined to the round flask and put onto the heating mantle. In the Gjeldahl method, the sample is digested in a heating mantle with concentrated sulfuric acid and catalysts at 350 to 400 degrees Celsius for about 1.5 to 2 hours. Heating continues until the mixture becomes clear, ensuring complete breakdown of organic matter and conversion of nitrogen into ammonium sulfate for further analysis. Now, the sample is cooled and diluted with 200 milliliters of double distilled water and made basic by the addition of 50 gram of sodium hydroxide. After the digestion of sample with sulfuric acid, the solution in round bottom flask is cooled and 50 gram sodium hydroxide is added to make the solution alkaline by carefully shaking, shown in first video. The basic nature of the solution is checked with litmus paper by dropping few drops solution with help of pipette on the paper, which turn to blue color. After the solution is transferred to Jaldahl flask for distillation of ammonia gas, shown in the image, and also the boric acid solution with bromocazole green methyl red indicator, pink colored, is prepared, shown in image, for absorbing the ammonia gas. This unit shows the whole Jaldahl flask for distillation of ammonia. The Jaldahl flask is connected to a condenser through a tight-fitting connector, which leads to a conical flask containing 50 milliliters of boric acid solution with a mixed indicator for ammonia absorption. It is essential to ensure there are no leaks between the Jaldahl flask and the conical flask to prevent ammonia loss, as this would affect accurate protein determination. During distillation, the solution color changes from pink to light green, confirming ammonia absorption. Distillation is continued until the volume of the distillate reaches about 150 to 200 milliliters, ensuring complete transfer of ammonia gas for precise nitrogen estimation. Next, during distillation, the color of the solution in conical flask changes from pink to light green, confirming ammonia absorption. Distillation is continued until the volume of the distillate reaches about 150 to 200 milliliters, ensuring complete transfer of ammonia gas for precise nitrogen estimation. Now, the solution containing the absorbed ammonia is titrated with 0.1 normal HCl solution, until the color changes from green to pink color. The volume of titrate value is noted for food samples. Similarly, the blank experiment is performed by taking pure sucrose instead of the food sample containing protein. Sucrose is a carbohydrate that contains no nitrogen, so it serves as an ideal blank material. During digestion with concentrated sulfuric acid and catalyst mixture, such as potassium sulfate and copper sulfate, sucrose decomposes without producing any ammonia. This ensures that any nitrogen detected during titration originates only from the reagents, not from the blank substance. Thus, the blank is carried through all steps, digestion, distillation, and titration, in the same manner as the actual sample. The titration value obtained for the blank, using sucrose, is subtracted from the sample's value to correct for reagent impurities and obtain the true nitrogen or protein content of the food sample. Finally, the calculation for determining the percentage of protein in food samples, which measures the nitrogen content and converts it into protein content. First, the percentage of nitrogen is calculated using the formula. Nitrogen percentage is equal to Vs minus Vb into the normality of acid and multiply by 1.4 divided by weight of sample, where the Vs is volume of HCl used for titration, Vb is volume of HCl used for blank experiment, N is normality of HCl, W is the weight of food sample. Here, the nitrogen percentage obtained is 0.896%. Since proteins mainly consist of nitrogen-containing amino acids, the nitrogen percentage is multiplied by a standard conversion factor, usually 6.25, to estimate the total protein content in the sample. This gives, protein percentage is equal to 0.896 into 6.25 equal to 5.6%. Therefore, the total protein content in the analyzed food sample is 5.6%. This calculation helps in assessing the nutritional quality of food products, as the protein content is an important indicator of food value, especially in cereals, pulses, milk, and other dietary sources. Similarly, the Jaldahl method can be used to determine the protein content in various food samples. Protein levels vary widely across different foods. For example, milk contains about 3 to 4% protein, and yogurt has 3 to 4%. Eggs provide 12 to 13% protein. Among legumes and nuts, boiled chickpeas contain 8 to 9%, raw soybeans are very high at 36 to 40%, peanuts 25 to 26%, and almonds 20 to 22%. Meat and fish sources include chicken, which contains 25 to 31%, and salmon, which provides 20 to 22% protein. Cereals and vegetables have lower protein content, with wheat flour 10 to 12%, cooked rice 2 to 3%, and leafy vegetables 2 to 4%. This data helps in estimating dietary protein intake. Thank you for watching the Analytical Chemistry Vision, and if you like such videos, please visit these sites for interesting videos for theoretical and experimental lectures. Also, there is video for determination of moisture, ash and fat content in food samples. Please click on like it and subscribe it. Thank you.